Good morning, everybody. So now, as we have discussed already several times, we will switch from the chemical stage to the biochemical stage of radiation, first with the DNA and then with the chromosomes. So the first part of this lecture will be about DNA damage and repair. And this is by far the most important, the starting of all biological effects of radiation. We will see the classical idea of how radiation has impact on organisms. That is the damage to the DNA. This we have seen already in the chemical phase. There are, however, many types of DNA damage that can be induced by all kinds of environmental stressors, including heat, heat, chemicals, and of course radiation. For radiation in particular, there are some types of damage like the thymidine dimers, the joining of the bases that are typical of UV radiation, not so much of ionizing radiation. But for ionizing radiation, you can have base damage and you can have strand breaks, either single strand breaks or double strand breaks. We will see that strand breaks are much more important than base damage. And the reason is that, as you can imagine, base damage, because of the complementarity of the DNA strands, base damage can always be relatively easily repaired because you know exactly what you have to fix here. But we will see that double strand breaks are probably the most important lesions. So a few words about the measurement of DNA damage. I mean, historically, it was quite difficult. Now it's becoming much more easy. But in the past, many data were based on plasmids and on gel electrophoresis in what was called the COMET assay. Now, by far, the most popular technique is immunofluorescence staining of the damage response protein. So you don't really visualize the DNA directly. You visualize the proteins that are bound to DNA and that become modified when there is a damage in the DNA. This is old-time example, not so old actually. This paper is from 2012, but this is done with electron microscopy and shows the damage to plasmid DNA. So it's a short string of DNA. It's only 2.9 kilobase pairs. So it's let's say it's shorter than the coronavirus, if you remember. That's some 30 kilobase pair. So it's like a virus DNA. And this is, you see, irradiation A, B, C, D are different doses. This is 1 kilogray, 3 kilograys, 5 kilograys, and 12 kilograys. And you see, of course, the chopping of the DNA. More and more you chop this DNA plasmid, increasing the dose. Traditionally, a very famous method to look at the DNA damage was gel electrophoresis. So essentially, you were putting the DNA in a gel, applying an electric field, because the DNA is an acid. So being an acid is negatively charged. The DNA will migrate. Now, if you have a plasmid, you know, when you can chop in pieces, every piece is a given size, you can have this kind of ladder. So every piece, of course, here you have the electric force and friction on the other side. So they will stop depending on the length. The big fragments will stop on top, and the small fragments will stop down here. So you can use a plasmid as a scale, as a ruler, and then after irradiation, you will have this kind of effect. You know, you will have low dose radiation, you will have this, and high dose radiation, you will have this. But of course, you need very large doses to see something, to measure realistically the DNA damage with this technique. A modification of this technique is the so-called COMET assay. The COMET assay you actually do gel electrophoresis of single cells. So you take a single cell and you exploit the fact that in the presence of the electric field and after radiation, the DNA is lost from the nucleus in small fragments. And you see, this is a situation in low dose, medium dose, and high dose. So if you measure the ratio, and this is in color, this is the same story, but in color. If you measure the ratio between the tail moment and the head, you have a correlation with the dose. So the Comet assay is still used sometimes for quantifying the dose, but all these methods has been now overcome by the H2AX assay, the H2AX assay. This is relatively recent. It actually, the first paper came out at the end of the 20th century, like 1998, I think, from Bill Bonner lab at NIH in US. This is in fact one of the first papers on Nature in 2003 describing the process. So essentially we have seen the DNA is wrapped. You remember in the biology, lecture, we saw that the DNA is very much wrapped around the histones. Some of these nucleosomes, where there are the histones, some of them contain a histone known as H2AX, others contain a histone known as H2A. Now, when there is a DNA break, so when radiation comes and induces DNA, actually not a single-strand break, a double-strand break. So you have to break the continuity of the chain of, these were called, if you remember the biology lecture, these were called beads on the string. If you break your necklace, you know, imagine it as a pearl necklace. To break the pearl necklace, you really have to have a double-strand break. You know, you have to break the string in two pieces. At this point, you start with the reaction of several enzymes. And in particular, you can have three proteins are crucial here: ATM, ATR, and DNA PK. We will see these proteins a little bit better, but ATM it comes from ataxia telangiectasia. ATM stands for ataxia telangiectasia mutated. So it's a gene that was discovered in the patients of ataxia telangiectasia that are very radiosensitive because they are missing ATM. In fact, ATM is a sort of guardian, and ATR is similar to ATM but works in SG2 phase. ATM works mostly in G1 phase. And DNA PK, the DNA protein kinase, is downstream of ATM. So ATM is a sort of policeman, and the first thing that you do is phosphorylate. So it's a phosphorylation, so it's a kinase essentially. It's a kinase enzyme, and this kinase is phosphorylating only, you see, in specific position where there is an H2AX histone. At this point, what you do, if you have a phosphorylated enzyme, you can use an antibody against this phosphorylated enzyme. This is an antibody that generally they come from animals, from rabbits. So you can develop an antibody which is specific to target, to bound the phosphorylated H2AX. And you can put a fluorescent marker on the antibody. Now you will be able to see all the phosphorylated H2AX. And we can say, okay, but you know, this string that you see here is extremely small. You see, we have here only 4 phosphorylated H2AX histones. How can you see them at a microscope? You know, a microscope, or even in flow cytometry. The point is that this is what you see in the microscope. The point is that instead, this is extremely big. Why is so big? Because actually, for some reasons not properly understood, not yet at least, the phosphorylation around the double-strand breaks takes about 1 megabase pair. So even if you make a small DNA damage, the whole string, about 1 megabase pair around the DNA lesion is actually phosphorylated. So this gives you this impact. You see, this is actually a picture I took from Markus Lobrich, who is professor at the University of Darmstadt, actually in biology, and is one of the top experts in this field. So I have a lot of slides from him, you will see here. You see, this is how the damage looks like. So you use a DNA double-stranded break. So you have phosphorylation only in the H2AX enzyme. Then you can use a primary antibody, and you can use, normally this is what you do. You have two antibodies, a primary antibody against the H2AX, and the secondary antibody with the fluorochrome against the first antibody. So you can amplify the signal. And that's how it looks like. You see, this is a nucleus of a cell stained in DAPI, which is a blue fluorescent stain that binds all the DNA. And then you see these points, these green points after radiation. Each one of these green points represents a DNA double-stranded break. And then you see, if you wait, you see that these green points disappear because the DNA double-stranded breaks are repaired. And this is the key point, you know. The key point in radiation biology is really to understand the repair, and not only radiation biology. DNA repair is one of the most important processes in so many diseases, including cancer, you know, or huge number of diseases. So to understand how the DNA is repaired is definitely a key step. So the damage, of course, but this we have already seen, the damage on the DNA strands, either single-stranded breaks or double-stranded breaks, can be either direct or indirect, depending on whether you have direct ionization on the strand, or you have actually ionization in water, and then the free radicals, especially the hydroxy radical, which this is the one with the higher G value, or the product like hydrogen peroxide or the ROS, oxygen fixed radicals, induce a damage into the DNA. Now, what is a double-stranded break? Double-stranded break is what we call from Shakespeare, the Julius Caesar, the most unkindest cut of all. That's, you know, when Caesar is, he receives this 33 stabs, but the last one comes from Brutus, and then he says this is the most unkindest cut of all. What is the most unkindest cut of all? It's a double-stranded break. And the reason, if you think about it, is pretty clear, because the base damage we see are easy to repair, but even single-stranded breaks are easy to repair, because, you know, it's a single-stranded break, so you can actually still have you can still fix the base, you have the complementarity, close here with the ligase. Typically, this is work done by enzymes called ligases. You can ligate the phosphate backbone, the sugar phosphate backbone, and that's repaired. But if you have two single-stranded break close enough, and when I say enough, it's difficult to quantify. We will discuss this a little bit more. Imagine that they are really not staggered. These are staggered. You see, these are staggered to, let's say, less than 25 base pairs. This is a common approximation, is to say that you have a double-stranded breaks when the two single-stranded breaks are about 25 base pairs away. But it can be less than 25 base pairs. Imagine that they are perfectly blunt. You know, that you have a cut where you have a single-stranded break here and a single-stranded break here. In that case, you see that you now have two strings. To rejoin these two strings is not so easy. So that's the most dangerous type of damage. So what is the dose effect? What is the dose response curve for DNA damage and DNA double-stranded breaks in particular? These are very old data. You see, these are data from 1996 obtained with the COMET assay. And looks like, you see, the doses are substantially high, but there is also some data at low dose. The impression is that you have a linear dose response curve. So the damage increase until you reach, of course, a saturation. Eventually, everything has to saturate, you know, but but this is very high dose. So the old data from gel already show a linear behavior. But we have to understand what is a DNA damage, though we have seen it. I mean, you see, in reality, if you look at the track, this is a Monte Carlo simulation, so it's not the experiment, but this is what happens if you take excitation. I'm sorry, here excitation is in, this is lost here, excitation is in green and ionization is in red. You see that if you take a track and you overlap the track to DNA, you can have, including the chemistry, of course, so including the production of free radicals, you can have a lot of events on the bases, on the strands, at different distance from each other. So what is a DNA double-strand break? Here I will follow the English school. I mean, this topic was studied very long at the University of Oxford by Dudley Goodhead and his students. So this is not universally accepted, but I will follow that scheme because I think it's very didactic, it's very clear for a student to understand. So we define the elementary lesion when we have a DNA strand break or a damaged base. So you see, this is a strand break, this is a base damage. This is an elementary lesion. If you have two or more elementary lesions within a few nanometers, this is generally about, as we have seen before, about 20 base pairs, you call this clustered DNA damage. If you have 3 or more elementary lesions, you call it complex clustered damage. So essentially, you increase the complexity of the damage. The idea behind the English school is that when you increase the complexity of the damage, the repair will be more and more difficult. And this is somehow intuitive, if you think about it. It's like having a dirty lesion. You know, instead of, if you have a string and you cut it with the scissor in a perfect way, then you can you put some tape and you repair. But if you kind of cut it in many different pieces, then it will be very difficult to rejoin it properly, like you see the difference between this case and this case. Now you see these are all relatively old papers, you know, from the end of the 20th century. The classification becomes more sophisticated even here, because of course now this is let's say the detailed situation, and this is a cartoon where essentially you see the two strands and the bases. So this is a case where you have no breaks. You see one strand, and all the bases are fine. This is a single strand break, okay? And we also agree on that. This is obviously a single strand break. This is what already we can call a SSB plus. We call it a SSB plus because there are now two single strand breaks. They are on the same side, so they are not a double strand break, but there are two of them. Now, if you have two single-strand breaks on opposite side at the distance of 20 base pairs, here in this cartoon is 10 base pairs, but you can imagine you can have 10, 20, 25. You take these numbers to adapt to the experimental data, essentially. Then, if it is, you see, if it is more than 10 base pairs or 20 base pairs, you call these two single-strand breaks. Because you see, if they are too far away, you know, if the two strand breaks are too far away, the cell will treat them as separated damage, not as one damage, but as two separated damage. But of course, if they are close enough, in this case, 10 base pairs, you will call this double strand break, you know, like in this case. And of course, if you have additional breaks, you will have a DSB plus, a DSB plus plus, and then of course you can add the base damage. The complexity will also be determined by the base damage. The idea is that if you increase the complexity of the damage, you increase the difficulty in repairing properly the DNA, And this means that if you increase the difficulty in repairing the damage, you will have more risk of late effects. This is now, you see, again, an example where now you consider also the base damage. You see single-strand break base damage, simple clustered damage where you have two components only. So now you have double-strand break, double base damage, and single-strand break plus base damage. Or when you have three or more components, and then you can have complex double-strand breaks and with the inclusion of base damage. So now you include, let's say, the complexity in the game. What is the fraction of clustered damage? This is interesting. You see, even when you take X-rays or gamma rays, about 20% of double-strand breaks are complex via one or more additional breaks. And 50% are complex via additional breaks or base damage. These numbers become 70% or 90% with high LET alpha particles. So, and you can imagine that if you go to high LET, we will see, of course, later, the complexity of the damage increase. But even with X-rays or gamma rays, the damage is fairly complex. This is due, as usual, to the sting in the tail, to the low energy electrons that can actually induce several ionization at the end. You remember the low energy electrons around the 2 nanometers, they have a very high cross section. So this is what happens, that when you are in the sides of 2 nanometers, even electrons can induce many ionizations and then can make many damage. That's why these numbers are somehow surprisingly high, even for low LET radiation. So the question is, how do we define exactly the complex clustered damage? You know, so when we have when we have two lesions, base damage and/or strand breaks, these are essentially simple clustered damage. But current experimental data to recognize, quantify clustered damage cannot distinguish between simple clustered damage and complex clustered damage. So somehow when the damage is complex, experimentally is very difficult to distinguish. The simulation says that a high proportion of clustered damage are complex, and the question is whether they are more difficult to repair faithfully. So the research challenge is to develop methods to quantify complex clustered damage and to assess their biological implications. We will see that now there are some experimental data that show that this approach is essentially correct. So if you have complex clustered damage, the repair is more difficult than if you have simple damage or simple clustered damage. So how many of these we have now with those? I mean, just to give you typical numbers, I mean, we have per gray, we have about 1,000 single-strand break per gray, and we have about 30, 40, maybe even 60 double-strand breaks. We have DNA protein cross-links about 50, and the complex damage, simple complex damage, like single-strand breaks plus base lesions are about 60. The most important part in the DNA study is not the DNA damage, but is the DNA repair, because as I said, if you can repair the DNA damage, then you solve all the problems. So the DNA repair is the big topic, not only for radiation biology, but in general for molecular biology. And it's a very complex process, of course, because you see, it depends on many factors. You have the repair process, but you have the cell cycle checkpoint activation. We have seen already that the integrity of the DNA is checked at the checkpoints like G1S or G2M. There is a damage tolerance. Not necessarily everything has to be repaired. I can accept some damage. There is apoptosis, so the damage can be so bad that I want the cell to die. This we will see in the next lecture. And the transcriptional activation, so to react to the damage, I have to produce specific proteins that then I will use for repair, and then I need transcriptional activation at the DNA level. So these are the examples. So to make it extremely simple, we will see many of these cartoons much more complicated. To make extremely simple, when you have a double-stranded breaks, the first key player is ATM, and ATM has a double role here. One is the phosphorylation of H2AX. We have seen this already. I forgot to tell you that the phosphorylated form of H2AX is called gamma H2AX. The phosphorylation of H2AX is the first step towards repair through many different repair pathways. This we will see. But ATM also activates P53, also actually phosphorylates P53 directly, and P53 is responsible for the cell cycle arrest and eventually for sending people to death, cells to death. I'm sorry, not people. So you see, in a perfectly working machinery, you should have a system where when you have a damage, either you repair it faithfully, following this way, or you kill the cell to avoid that it can make damage in the future. How do you see the repair? Of course, you can measure the rejoining. So you can see how many pieces you have at the beginning and how these pieces are rejoined. These are, again, very old data from gel electrophoresis in the 20th century, before the discovery of H2AX. But the shape of the kinetics is similar and is generally a double exponential. It's not a single exponential. Can be sometimes approximated with a single exponential, but to be precise, is a double exponential. You see, with the fast repair, with a half-life of about 10 minutes, and then a long repair with a half-life of about 3 hours. It's 3 hours, 180 minutes. So it's a double exponential function that gives you the rejoining of the DNA, and the rejoining, of course, means repair. Not necessarily correct repair, though. You can imagine that you can rejoin the pieces in an incorrect way, and you will not see this with gel electrophoresis. Actually, you don't see it not even with the gamma H2AX. You have to sequence the translocation to see that, which is something which is now some groups are trying to do. So what are the repair mechanisms? The repair mechanisms are really fascinating. The complexity of the DNA repair mechanism probably shows you how important they are, how the evolution of this mechanism made it so complex. We see here two typical, very simplified cartoons. We will see them in the real life, let's say. This is the excision repair. Excision repair is used for repairing single-strand break, you know, when you have a single-strand break. So when you have a single-strand break, you already need a protein, a complex of proteins that works. You have to re-synthesize the bases that you remove when you include this protein complex, and then you ligate. The last step is generally ligation, which is done with a specific enzyme, which is known as DNA ligase. For the double-strand breaks, the procedure you can already see here is extremely simplified, but is much more complicated. And for sure, one of the key roles, we have seen this already, comes from DNA PK, because DNA PK is the protein that stabilizes the broken pieces. So now you see here, from the physics point of view, the molecule is stable from the physics point of view. It's stable because, you know, even though you have now a broken piece, the molecule still has its structure, you know, but here the molecule is unstable. So in principle, the thermal fluctuation can bring the strands away the original configuration. So you need a protein which is DNA PK. In particular, DNA PK is a complex too. These two proteins here are the Q complex, Q70, Q80, that keeps the two strands together. If you keep the two strands together, then you can actually repair the DNA double-strand break. That's a little, and there is a further complication here that I will show you in a second. You see, this is a little bit more detail, still not the full story, but a little bit more detail, and especially shows you that there are two mechanisms that can be activated. One mechanism is the so-called non-homologous end-joining. And in the non-homologous end-joining, you see, you induce a DNA double-strand break. ATM is the policeman, the sensor that phosphorylate the H2AX histone, and there is accumulation, eventually there is accumulation of other enzymes like Artemis, especially if it is a complex damage. It's not always recruited, but if it is a complex clustered damage, then you will have Artemis. Then you can go through the so-called non-homologous end-joining. For non-homologous end-joining, you see, you need something to stabilize the breaks. And these are the Q70, Q80 complexes. These are small proteins that bind the free ends. The DNA PK that keeps them together. And then at this point, you start with a large number of other proteins that will actually rejoin. The final step is always a ligase and will rejoin the piece. But you see, this mechanism, the non-homologous end-joining, is relatively simple, but relatively error-prone. Because you see, you essentially, you try to keep the piece together and you try to rejoin, but if it is a complex damage, unavoidably, you will lose some piece of DNA. So you rejoin something which is not exactly the same as in the other case. You can do better if you have a template. And if you are in S phase, especially late S phase and in G2, you have a template because you have two DNA copies, because if you go through the S phase, you have a perfect copy. If you have a copy, then you can use another mechanism, which is known as homologous recombination. In homologous recombination, you see, well, it's also a very complicated system. Essentially what you do, you have your broken DNA, and now you invade the homologous chromosome to rebuild the exact sequence that was broken. This is in a simplified way. We will see that it's more complicated than that. The proteins involved are also different. There is ATR instead of ATM. There are other proteins like RAD54 and BRCA2, which is involved in breast cancer, that are not involved in non-homologous end-joining. So it's a different mechanism, and is very accurate, but is also very slow. So even in SNG2, the majority of the damage is repaired through homologous with non-homologous end-joining. Also, the choice between non-homologous end-joining and homologous recombination very much depends on the complexity of the damage, with a preference for homologous recombination in SG2, of course, because you cannot have homologous recombination in G1, with a preference here when you have complex DNA damage. So you see, here is a little bit more on the homologous recombination pathway. In the homologous recombination pathway, the first point must be that you must have the two strands. So you have to be either in S phase, better in late S phase, you know, when the replication is completed, or in G2. You cannot be in G1. Then you have, of course, you must have a double-strand break. And the first player is always ATM. And then ATM recruits new protein, like MBS1, MRA11, RAD50. These proteins you see here, they start a cleavage of the broken DNA. You see there is a real cleavage here. Many proteins involved here. At this point, you have changed drastically the initial configuration of the strand breaks. You see now you have cleaved a lot of bases here, and you have two sticky ends in the DNA. And now you have to nick the other DNA, and you have to invade, and this is mostly done by two proteins, RAD52 and RAD54, that invade the other strand. With the invasion of the other strand, you can actually reconstitute completely the original strand. Many proteins, many proteins, and an amazing complexity. These proteins called RAD, there are many of these proteins called RAD. These are all discovered in yeast. Actually, homologous recombination is used in yeast for DNA repair. So these proteins were actually discovered in yeast, and it's quite remarkable that the same protein used by yeast that are, of course, you use yeast to make pizza. You know, it's a very simple organism. That same mechanism is conserved evolutionary up to humans. We use the same proteins, we use the same mechanism. We repair our DNA like yeast. Also, some other proteins are also familiar. For example, BRCA. BRCA stays for breast cancer, because mutations in these genes predispose very much to breast cancer. So much that actually, if you have a double mutation in BRCA1 and BRCA2, the probability that you get breast cancer is essentially 100%. You remember Angelina Jolie? Angelina Jolie decided to have a double mastectomy, even though she was fine, the famous actress, because she knew that she had this mutation in the BRCA gene. So the process of homologous recombination, you understand, is not only important for repair of radiation damage, it's also important in general for the correct repair of DNA. And if you cannot do a correct repair of DNA, you can have cancer. Let's go back for a second to the complex clustered damage, because we have seen here Artemis. Artemis also is a protein discovered by Penny Jago in UK, together actually with Marcus Lobrich here at the University of Darmstadt, in our own university. And it's a work in progress. So I don't give you, let's say, final answers here, but you see how important it is. If you have moderately complex double-stranded breaks, then you need Artemis in the non-homologous end joining repair, and you find that approximately 10% of double-strand breaks require Artemis after gamma rays, and 20% after alpha particles. And you can also do synthetic constructs. So instead of inducing by radiation, you induce the damage with specific enzymes. With the three-component clustered damage. So you make a dirty double-strand break. Dirty means clustered in a way. And these actually are more difficult to repair. So the idea that they are more difficult to repair is somehow justified. It's also a work done in Oxford, in Harwell, which is correct to Oxford, showing that the complexity translates in a more difficult repair. Again, these are now not DNA repair proteins. These are actually DNA damage checkpoint proteins. So these are the proteins involved in the checkpoint. But many of them are essential for repair, of course. Checkpoint and repair are strongly related. And this slide, of course, I don't want you to remember these names, but is only to show you again that these are mammalian cells, and these are two yeast, Saccharomyces pombe and Saccharomyces cerevisiae, and you see how many similar, I mean, sometimes they use different, like ATM and ATR are only for mammalian cells, but you see that the RAD proteins are present also in yeast, and even the transducers kinase, those that essentially that allow the transition in the cell cycle, like checkpoint 1 and checkpoint 2, they are also in yeast, you know. This is how now a more realistic cartoon. I will not go into that. If you want to have fun, try to go now into the detail on the, and this is, you see the progression in the cell cycle and how the DNA damage and ATM, which is now we have is the first and most important protein that plays the role here is ATM. From here, how many steps you have toward either double-strand break repair or cell cycle checkpoint blockade. And that's a little bit a simplified cartoon of the checkpoints. You see here, this is G1, S, and G2M. So this is essentially what happens when you have a G1S block, a block in G1. You see the double-strand break, ATM, recruitment of many different proteins, MDC1, 53BP1, MRN, BRCA1, P53 phosphorylation that can be actually inhibited by CHG2, P21, which is phosphorylated by P53, and eventually the block in G1S. Similar things happen, you know, in S phase and in the G2M checkpoint. Here, the role of ATM is actually taken by a protein which is very similar to ATM, but it's called ATR. It's a slightly different version of ATM, which is activated only when you have when you are in S phase or G2M. But you see the process again is the double-strand breaks, phosphorylation of CHEK1 or CHEK1 and CHEK2, involving of other proteins, and eventually inhibition of the transition. So you see the checkpoints, the message is that checkpoints in DNA repair are very much entangled, very much entangled. Now, I told you that essentially there are two main pathways for the repair of double-strand breaks. And these two main pathways are non-homologous end-joining and homologous recombination. Non-homologous end-joining tends to be error-free. Homologous recombination tends to be, non-homologous end-joining tends to be error-prone, and non-homologous and homologous recombination tends to be error-free. But apparently there is even another pathway that can be activated when the cell is really unable to repair the damage. You see, as I said, you have a double-strand break. If you go through the non-homologous end-joining, the so-called canonical non-homologous end-joining, one of the first steps that you need is the Q protein. The Q protein actually acts as a, probably, not for sure, as an inhibitor of the CTIP. CTIP, also called RBBP8, is an end resection nuclease. So it's a protein that cuts the strand that makes, you see, that makes sticky ends. If Q is able to bound the strand breaks, then CTIP is inhibited, and you will proceed through the canonical non-homologous end joining. Not necessarily this will be perfectly correct, but will be pretty good. We have seen, I mean, it's a process where essentially at the end, you keep the breaks there, you have other proteins coming, and then eventually you ligase. What if you cannot do it? What if Q is unable to bind because the break is too dirty? You know, because you have too many, imagine that you have too many breaks here, too many base damages all together. The damage is too clustered, the damage is too dirty. Then Q is unable to bind, so Q is unable to inhibit CTIP. Then CTIP comes, and CTIP will make resection. Resection means that now you cut long strands on the two sides until you find a microhomology. So until you find sequence 2 or 3 base pairs that are the same on the two sides of the strands. At that point, what do you do? Boom, you actually join the microhomology and you repair the DNA. Of course, this kind of repair is extremely error prone. For sure, here you're making a large error. You are changing the sequence completely. It's called alternative non-homologous enjoining. So when the cell is unable to use the normal one, it goes through alternative non-homologous enjoining. And you understand that this can be done involving, if you have more chromosomes broken, like in this case, you see you have red, blue, you have different chromosomes broken. This can lead to translocations. This can lead to joining chromosomes that should stay separated. But if you go in microhomology, if you break different chromosomes, you join whatever is around. So this alternative non-homologous enjoining is now attracting a lot. It's fairly recent. You see, this paper is from Maria Jasin at the Memorial Sloan Kettering in New York, is 2011. So it's coming quickly. And that's, you see, another example. Let's say that how from the double-strand break you can have homologous recombination. This we have seen, but only if you are in SG2. You can have the classical non-homologous enjoining, or you can have, involving CTIP, the alternative non-homologous enjoining with the end processing of microhomology and eventually always ligation, of course. There is a lot of discussion, on this point. I mean, whether resection-mediated alternative non-homologous enjoining is really the cause of chromosomal aberration, cell killing, of all the, let's say, everything we see as a consequence of the damage is not really due to unable to repair, to the fact that the cell is unable to repair. If the cell is unable to repair and the P53 is active, this should lead to apoptosis. But if you have a misrejoining, so if you have a rejoining of the DNA which is inappropriate, this can lead to chromosomal aberration and cell killing. So you understand that alternative non-homologous enjoining is definitely a big topic. Now, there is a discussion. For example, some authors think that alternative non-homologous enjoining backup enzymes are filling in when one or more primary non-homologous enjoining components are mutated, missing, or overwhelmed with a large number of very complex double-strand breaks. For example, when you have heavy ion. So essentially, you see, instead of having two different lines, one is non-homologous enjoining, the other one is alternative enjoining, you can have a mixed situation where in some cases, when you have, for example, many breaks, or when you have a mutation, this has been seen in cells that have specific mutation in specific proteins for non-homologous enjoining, then you can resort to some proteins in alternative non-homologous enjoining. So it's not necessarily two different pathways like it looks here. If you look at these slides, looks like you have two pathways that are separated. In reality, it could be, some proteins can come from here when here you have problems. As I said, a lot of these studies are done using repair deficient cells. So you take cells that are specifically deficient in some of this protein repair. In some cases, especially these days, you build these repair deficient cells. In other cases, you just, especially with the modern techniques of CRISPR-Cas9, we didn't have time in the biology lecture to go into this technique, but you know that these days in genetic engineering, you can engineer essentially everything using CRISPR-Cas9, which is a technique where you can actually move a gene in a specific position of the genome. But in the old time, like here, this is an old paper done with the electrophoresis. This is the fraction of damage of DNA damage as a function of the repair time. You see the double exponential behavior. The SCID, for example, SCID is a disease which is a deficiency in non-homologous enjoining. So the cells that are deficient in non-homologous enjoining, you see that they have problems in repairing. They are, instead of going down the rejoining here, they have problems in rejoining. And there is Fast rejoining is still working, maybe in alternative non-homologous joining, this we are not sure, but there is a fraction of unrejoined cells. And the consequence of these DNA repair genes are generally very gloomy, very bad. You know, these are example of Xeroderma pigmentosa, which is probably one of the most common DNA repair defects. This is actually not a defect in DNA double-stranded break. Is a defect in nucleode excision repair. And these people cannot really go into the sun because, of course, because of the UV, they will be immediately burned, and they have a strong effect of the UV light. You see here a number of, fortunately, much more rare disease involving different genes. You see, ataxia telangiectasia is probably the most famous because the gene responsible for ataxia telangiectasia is probably the most important gene in DNA repair, the ATM. Most important, it's the first, you know, it's the first in the chain. And you see the phenotype is very bad. You have immunodeficiency, neurodegeneration. You see how DNA repair is related also to neurodegeneration, and of course, cancer predisposition. Then you have other syndromes, ATLD, MBS, Seckel, Fanconi anemia, and so on, that are related to specific defects in genes involved in DNA repair. We have seen some of them, MR11, MBS. The MBS is a Nijmegen breakage syndrome because it was discovered in Nijmegen. BRCA, we already saw that is also involved in cancer predisposition, and in Fanconi anemia. SCID, actually SCID that we have seen before is a problem in the Artemis gene. And Lig4 syndrome is a problem in ligase. You are unable to make the final step of ligation of DNA. Now in the second part, we will focus a little bit more on the visualization of the data of the DNA breaks and how we can actually see this damage, and how comes that we can build these cartoons. You know, how do we know which protein is coming first and which protein is coming after? We will see this in the next part of the lecture.